the medial geniculate nucleus, which is involved in the auditory way station in this cortical loop down into the subcortical area. So it's a cortical-subcortical feedback system, and they're kind of distinct areas or nuclei for the different sensory systems. Well, what's interesting is within these different nuclei, there are two basic types of cells. There's one set of cells which are called the magno cells, and they're called magno because they're large cells. And then there's the parvocellular system, and those are smaller, more compact cells. The magnocellular system are larger because they're more myelinated. And so one of the theories is that the magnocellular system is involved in the transient transmission of information, the rapid transmission of information, as well as a variety of other features. And so work of Al Galaberta at Harvard, who's done the only neuropathological studies of the brains at the anatomical level, the cellular level, of the brains of humans that had been known to be dyslexic before they died, and then donated their brains for science, and they were able to really study them. Al Galaberta's work had shown that there's a disproportionate abnormality in the magnocellular systems in both the LGN, the lateral geniculate nucleus of the thalamus, and the MGN, and the medial geniculate nucleus. And that, therefore, translates back into a physiological, neuroanatomical, neurophysiological basis for a difference in the processing rates that those brains might have been able to sustain. And so Al Galaberta is a really good person potentially to talk with, because he's developed a whole series of animal models to look at what are the effects of having deficits in the magnocellular system, and also what are the connections between the magnocellular regions of the thalamus up into the cortex. And then John Stein at Oxford has also really developed this magnocellular deficit hypothesis, which is one of the focuses right now on dyslexia research at the more physiological level, and it fits very, very well with the temporal spectral processing deficit. In fact, many of the current theories, be they phonological processing deficit or whatever, they have in common that there seems to be some underlying difference in the processing rates of the individuals, or difficulty in processing rapid transient information. And John Stein's lab, Joel Talcott and Carolyn Witten, and various other people in John Stein's lab at Oxford, have published some wonderful papers in the Proceedings of the National Academy of Sciences recently, I think in 2000, that showed that really fit well with the study I talked with you about with infants. Basically showed that if you look at just normal healthy kids, if you look at a whole classroom of children, let's say in England, who, different levels of reading ability, you've got the whole spectrum of individual differences, that you find that the ability to process transient visual and or auditory information is very highly correlated. The visual transient processing seems to be highly correlated with orthographic abilities, and the auditory transient processing problems seem to be, abilities, seem to be highly correlated with the phonological decoding abilities of children. And this is across the spectrum, so it's very nice.
And implicate in both is the core processing frequency and
yes.
the ability to make distinctions.
Exactly. That's what it's all about. It implicates the core processing frequency and the ability to make distinctions, both in the auditory and the visual modality. And then how those come together. So they're highly correlated with each other. And what was really interesting in the Talcott et al. Paper was that they showed that just the ability to process transients in non-linguistic stimuli. So they're now in this, they were talking about modulating tones and the rate of modulation, frequency modulation within tones, and what the threshold is for hearing a wavy tone versus a straight tone, as it were. Because when it goes woo, when it has woo too fast, all of a sudden you start hearing a solid one. And just for normally developing children, your threshold, the individual child's threshold on that, was as highly correlated with their phonological awareness, phonological decoding abilities, as their reading and spelling were correlated with each other. I mean, it's really very high levels of correlation.
We're going to see at some point little new kinds of exercise toys for children that just draw out exercising the frequency of distinction in a more implicated general way to the various senses that are engaged in their play.
Right. Well, I mean, that is, of course, one of the underlying ideas of the fast-forward training programs, was therefore to say, well, if these processing rates seem to be so fundamental and so highly correlated, both predictive correlations from infancy to childhood and also concurrent correlations throughout life with your phonological abilities, your language abilities, your reading abilities, then is it possible to drive these rates of processing to make finer and finer distinctions in the brain? And so we can use computers to basically do those experiments. And we have done so and found that, in fact, it's amazing that one is able to drive processing rates quite substantially across the spectrum. You can make just about anyone a faster processor, but it can be very important for the individual who might be struggling with language and literacy skills because their processing rate may have been slower at a certain point early in their life where it was important. And you can drive them to faster rates of processing and then look to see whether or not language subsequently and reading skills subsequently improve, and voila, they do.
It seemed to me that there would be two fields that this would be applicable. One would be to having a faster frequency so as to be able to make the distinctions in time, so whatever the variation is in what's going on in the world. The second one is to have a higher frequency with which to go through the disambiguation that's involved in assembly, because on the one hand we've got these distinct elements which are required. On the other hand, before we get to wherever we're going, we've got to assemble them.
That's right, back together.
They have to go back together in some way, in which if there's ambiguity in the distinctions, it has to be processed through in the assembly. So the more that there's ambiguity in between that, the more there's time consumed in working it out, the more. So our approach is to increase the speed of the processing and wherever possible decrease the unnecessary ambiguity that's overwhelming the process. Right,
and that is exactly the same that is exactly the coming at the problem from both ends that we've used in the development of Fast Forward. Exactly, you got it. That's exactly what we tried to do. Both increase the processing capacity and also decrease the ambiguity in the signal itself by acoustically altering it so that these rapidly occurring frequency changes over time are slowed down and amplified. And then as the individual progresses in the various levels of language and language to reading and reading, one can begin to drop out these additional cues while at the same time having rewired literally the processing constraints that the kid had to begin with. How do we know that we really have literally, quote unquote, rewired it? Because we now have the advantage of technological imaging, in non-invasive imaging in humans, both through evoked potential electrophysiological methods and also through functional neuroimaging methods, both of which have now been done, magnetic imaging methods, and have been able to show that indeed not only are the children behaviorally processing more quickly, their language and reading scores on standardized tests are improving, but the differences between their brains and the brains of children who are better readers are becoming less distinct over time. So those areas that are more involved in certain aspects of the reading task, particularly the phonological aspects, which are distinctly different between a group of children who are struggling with reading and a group of children who are reading, really well. Over time, if your methodologies of intervention are working, one should be able to determine that using not only behavioral methodologies, but also physiological and neuroimaging. And so that's a real advance. I mean, this is brand new data. For the first time, we're beginning to see studies in which one is able to actually evaluate the effectiveness of reading interventions or other types of interventions. This is very similar kinds of approaches are being used with patients with stroke, both in the motor system, in the language system, in the perceptual systems, sensory systems, and particularly it's going to be, we'll see more and more studies coming out in which these neuroimaging procedures are being used as assessments for the efficacy first of all, assessments for a deficit, and then used as an way of evaluating, an additional way of evaluating efficacy of one method over another. So it's